Hello everyone, today we are going to talk about QSAR methodology and its importance. What is QSAR modeling? Drug development requires high time and money investments. Sometimes, it also needs in vivo testing, which raises ethical considerations. Besides, it's estimated that only 1 from every 9,000 active biological molecules reaches clinical use. To help in the drug development process, we have computational methods, and one of the most common is QSAR. QSAR stands for Quantitative Structure-Activity Relationships and is a methodology that looks for a correlation between properties, such as physicochemical or toxicological properties, and the molecular composition of the molecule. To achieve this goal, datasets with data coming from experimental assays are used. With this data, a mathematical model linking the analyzed property with the structure of the molecule is done. Later on, this model is used in order to predict the properties in new molecules. The workflow for the creation of a QSAR model usually consists of 7 steps as follows. 1. Database cleaning. 2. Descriptors calculation. 3. Feature reduction. 4. Creation of the train and test subsets. 5. Descriptor selection. 6. Model generation. 7. Model validation. Step 1. Database cleaning. To avoid problems in the following descriptor calculation, we take some measures. Remove the counterion in the salts. Remove the water excipient if exists. Remove incorrect molecules. Remove inorganic molecules. Remove organometallic compounds. Evaluate the mixtures. Evaluate duplicate molecules in the dataset. Step 2. Molecular descriptors calculation. The descriptors used to build up the QSAR models could be classified into different families, including simple structural features, such as number of heteroatoms, chlorine, etc. Fragments and substructures, such as number of carbonyls groups, amide groups, etc. Physicochemical properties, such as molecular weight. Topological indexes, taking into account the flat structure or molecular graphos. 3D descriptors, depending on the three-dimensional structural conformation. Step 3. Feature reduction. It's important to reduce the number of descriptors, removing the ones that don't provide information. This means removing descriptors with a unique value, descriptors with a number of missing values above a threshold, correlated descriptors. Step 4. Split dataset. The whole dataset is then split into 2 subsets, one to train the model and other to validate it. The training set usually contains between the 65 to 80% of the whole dataset and the training set the remaining compounds. Step 5. Descriptor selection. Among all molecular descriptors, those that are most correlated with the experimental values should be selected. The ones that best represent the study property for the training group are selected using different statistical filters. Step 6. Model generation. The number of algorithms and statistical techniques useful to carry out QSAR modeling is very wide, and their selection depends on the nature of the dataset and if the prediction is quantitative or qualitative. These algorithms can be classified by families, where the most commonly used are linear algorithms, neighborhood algorithms, supporting vector machine algorithms, tree-based algorithms. Step 7. Model validation. The last step implies the validation of the built model using the test set that we've saved it earlier. This allows us to check if the model works also with a set of molecules not involved in its training. The kind of metrics used to appreciate the goodness of fit depends on the model. In regression models, the most used metrics are R2, the mean absolute error and the mean squared error. For classification models, usually we rely on the confusion matrix and its related parameters as accuracy, sensitivity, and specificity. But, after all the effort in order to build up a God model, it is valid in the eyes of regulatory agencies? The answer is yes, but only if it follows some rules. 1. The experimental conditions should be the same for all the data points contained in the dataset. 2. The algorithm should be unambiguous, being reproducible for the scientific community. 3. The applicability domain should be well defined, so the QSAR model can be applied only to predict on compounds from the same chemical spaces as the molecules used to train it. 4. The metrics of the model should be properly reported. 5. Only if possible, we should try to supply a mechanistic interpretation of the endpoint. That was all for this video. Thank you so much for your time. You can contact us by visiting our website or our social media on Twitter and LinkedIn. We hope you enjoyed our video. Goodbye.